Well, hello, everyone. It is great to see you. Happy Friday if you're joining us live here. This is, again, part of our 2022 Research for the Public Good Conference at APUS. And we are very happy to have here this morning Lori Byerly, Kristen Ondrak, and Karen Golovin talking about their recent research project. And so I'm going to turn the floor over to them, and I'm so excited to hear about the work that they've been doing.
There we go. I think everyone can see that. So hi, everybody. Thanks so much for being here. Happy Friday as well. So as Jenny said, Dr. Byerly, Dr. Ondrak, and myself are here again. It seems like we are a team that just keeps on going. We've been here before. We'll probably be here again. We talked about actually naming our research team, so we would entertain team names if you want to put those in the chat for us as well. This presentation is centered on a research study that actually began with a 2021 faculty research grant. You know, it's no secret that folks are trying to lose weight, build muscle mass, and often combine resistance or strength training with an increase in protein intake. There's some research that's reporting that 40 to maybe even 100% of athletes use some type of supplement, often protein supplements, intending to improve their athletic performance. You've probably heard this, I'm sure. Even more, many of you have probably thought about your own protein intake for one reason or another. And even though we know that those wanting to build muscle frequently consume protein supplements, there is really very little known about the effects of protein supplements on the gut microbiota. But before going any further, we have a question for you, and you can please answer in the chat. I can't see the chat, so Lori and Kristen, if anything comes up that I can address or you want to just let us know, just call it out. So the question is, how much dietary protein do you think you need each day? My chat disappeared when I started sharing, so. All right, and so we're not going to necessarily come back to that and answer individually, but here's an idea of your protein needs based on your body weight. You'll see that one more time.
We have some good answers in the chat, Karen. Some people are listing grams, some people are listing 1% of their daily calories, and some people are noting it depends on their body weight.
All right, look at that. So see, it's crazy. You know, we're doing research studies, but this is just across the population. A lot of people are paying attention to protein intake, again, for a variety of reasons. So the study we're reporting on today compared the gut microbiome and metabolome of self-identified muscle builders who either did or did not report consuming a protein supplement. Our participants actually were recruited from our sports and health sciences program here at APUS. All right, and next question. Again, please answer in the chat. Do you think that you get enough protein? And as you're answering, there's a comparison of one serving of protein. How many people eat that size of meat in this case? And then here's some examples of what a serving is. How are our answers?
Mixed. Some say yes, some say not always. Some say too much.
There we go. All right, one last question for you. Do you currently supplement with protein? So do you have some sort of protein supplement, powder, liquid, or otherwise? Again, please answer in the chat. As you're answering, Lori Byerly will be talking about our results next, and then Kristen will talk about our next steps. And so as the answers continue to come in, here's another look at your daily protein needs. Okay. And maybe before we go over to Lori, is there anything in the chat we need to look at, Kristen?
No, well, there's some great examples of and some questions I think we're going to answer coming up. So I may punt those to what Lori's going to cover. Okay. A lot of people say, no, they don't necessarily use a protein supplement, but they may try to get additional protein. So I think we'll clarify some of that.
Nice. Okay. All right. And I know we will come back to some of those. All right.
Okay, so I'm up, and there was one question here in the chat. Who was it that asked? Leslie asked whether protein drinks with 30 grams of protein is too much to consume at one time. So unfortunately, the answer is going to be, it depends, because it depends on what context you're taking that and when you're taking that. So sometimes with our muscle builders, so individuals who are out there building a lot of muscle and they've spent a couple hours doing a lot of resistance training, that 30 grams may be okay. We generally are recommending somewhere like 20 grams each meal. So that gives you an idea. You're not too far off with the 30 grams. Okay, so I'm going to talk about our study and tell you what we found. And what we did is we collected fecal samples from people who self-reported as being muscle builders. Now we had a couple ways, I'm not sure if you can see my pointer in your screen or not. We had a couple ways to check on whether they were actual muscle builders. We had a physical activity questionnaire that we gave them, and we also used the International Physical Activity Questionnaire. This is a validated questionnaire, but it does not focus on muscle building kinds of activities. So that's why we had this additional activity questionnaire. I'm pretty certain that we were pretty good at getting people who were really building their muscle, but again, it was all self-reported or self-identified. We also collected 24-hour recall information from them. You'll see those results. And we also had a protein supplement questionnaire that we used because this ASA 24 does not ask about specific protein supplements. And we wanted to divide, as you can see, the participants into ones who did not report using a protein supplement and those that did report using a protein supplement. And then finally, we just collected some general demographic information from the individuals as well. Oh, so do I need to do something with my voice? I see. Okay, so everybody, most people are saying it's okay. Hopefully you can hear me. Next slide, Karen. So this is our inclusion and exclusion criteria for participants. Because we were collecting a fecal sample, we had to be very careful that we got individuals who hadn't done anything that could impact their GI tract. And so for the exclusion criteria, you'll see there's things like laxative, diarrhea inhibitors, prebiotics and probiotics. Those had to be at least a week out from taking any of them. So if any of you think that those have lasting effects, here's some proof that they don't. Antibiotics, we wanted it to be at least 3 months since they had antibiotics. Prescription medications, the only one that we really allowed was oral contraceptives. The rest, if they were taking any others, they were out. And then, of course, they couldn't have any diseases of the GI tract. So things like cancer, irritable bowel disease, Crohn's disease. We were also avoiding people who are tapering. So if you're familiar with this group of individuals, then you might be aware that sometimes they will change their diet because they're in the process of really wanting to ramp up their muscle building, and then they will taper after that. The participants had to be 25 years or older. So in this case, fortunately, because we used students from APUS, most of our students are not the typical college age 18 to 22, but we wanted greater than 25 years. And of course, we wanted them actively building, and we did not care if they were male or female. What about diabetes? They would not have been diabetic or hypertensive because their list of medications would have ruled them out. And we would have seen that with the medications, we would have seen that diabetes and those types of medications. Next slide, Karen. Here's a breakdown of just some subject characteristics. There was no significant difference between those that reported using protein supplements, and that's going to be when in the next slide, you'll see PS and no PS, and PS are the ones that are reported using a protein supplement, and no PS are ones who did not use a protein supplement. And we ended up having a pretty equal number between the groups. There's no difference in age, weight, height, BMI, and body mass index that is, and I'll come back to the last two at the end there, and between the groups, we had a pretty good even split between males and females between the two groups, although it wasn't half and half. Physical activity, there was a significantly higher MET minutes per week in the group that consumed the protein, said that they consumed protein supplements, than the ones who did not. I do want to note, even though this is significantly different between these two groups, they're both at a very high level of physical activity. Bristol scale is a way that you measure the consistency of the fecal matter, and it's on a scale of 1 to 5. There was no difference between these, and what we're doing is 3 is pretty much right in the middle. One end would be diarrhea, the other end would be constipation. So their fecal matter was of good consistency. And this is supplements, the last one, no difference in the amount of supplements, and these are going to be supplements that were not protein supplements that we're talking at here. Next slide. And the next two slides are going to talk about their diet. And so there wasn't any significant difference in the number of foods that they consumed each day. There wasn't any significant difference in energy and carbohydrate intake between the two groups. There was a difference in protein and fiber, with the group reporting that they did use protein supplements to augment their muscle, consuming more protein than the group that did not use protein supplements. And the same true for fiber. The group that consumed protein supplements had a higher fiber intake than the non-protein supplementing group. I do want to note, though, that this fiber intake is still below the dietary recommended intake for fiber. So they aren't either group getting an adequate amount of fiber in their diet. I'm going to go back to the protein for just a minute. And we are above the values that Karen showed you early on there were based on 0.8 grams per kilogram. That's 0.8 grams of protein per kilogram body weight. And here we're sitting at much higher than that. You can see the group that didn't use the protein supplements consumed 1.5 grams of protein per kilogram body weight, where those that reported using protein supplements consumed over 2 grams of protein per kilogram body weight. I will tell you that this is with sort of at both ends of what we would recommend for this group to be taking for protein if they were actively building their muscle as they reported. But we still have an excessive amount of protein intake here. Next slide. Okay, folate was significantly higher in the group that reported using protein supplements, as well as they ate more intact fruits of citrus, melons, and berries. They ate more beans and peas. That's peas as legumes. So, and you can compute, or you can put beans and peas in either the protein food group or the vegetable group. And so in both cases, it's significantly higher in those that consume that protein supplement. Healthy Eating Index is a measure of how well their diets met the U.S. dietary guidelines. And there's no significant difference between these two groups, but there was a tendency for those that consume the protein supplements to have a higher intake of, to have a better HEI, Healthy Eating Index. But I'll tell you, if since we teach classes and we know our students, if they got a 62 out of 100, and this scale is 100 is the best, you'd still be flunking. So we're still not doing good on that. No difference in water, alcohol, or caffeine intake. Let's go to the next slide, please. Okay, now we're going to talk about the gut microbiome, and I want to explain that term for you. When we use the term microbiome, we're referring to the genes that are present in the fecal matter that we measured. In this case, we're going to be measuring the genes of bacteria. So we use 16S RNA to look at this. The genes, the microbiome, can be used to then identify the microbiota, and biota refers to the bacteria. The study sample size was 22 for the group that was said reported protein supplements and 17 for the group that did not report protein supplements. I see that in the so this slide looks at beta diversity. So we have hundreds of microbes or bacteria present in our colon. The colon is going to contain much more bacteria than the small intestine, the stomach, the esophagus, and the mouth. But there are bacteria throughout our whole GI tract. It's really when the food gets into the colon that the bacteria that are present there are going to multiply and really break down all of that food that enters it there. So you can look at the diversity of the bacteria that are present in the gut, and this is looking at beta diversity. So here we're looking at the diversity. So how many different kinds of species of bacteria are present in the fecal sample from those that did or did not consume the protein supplement is what we're looking at here. And in subsequent slides, the green will be the group that reported using protein supplements, and the yellow will be those who did not report a protein supplement. And what I'm showing here is that there is a significant difference in the beta diversity between these two groups. And so the group with taking the protein supplement have slightly more diversity than the ones that did not. Next slide. So we can take those genes that we use to identify the bacteria, and we can also look at what metabolic pathways, because those genes identify certain genes which you're familiar with that are associated with metabolic pathways within our body, but in this case, we're looking at it within the bacteria. And so you can see on the left here, we've got nucleotide metabolism and about 10 different pathways there. And on the right, it will show you. So if it's orange, then it's going to be more predominant in the non-protein supplementing group, whereas it's green, it's going to be more dominant in the protein supplemented group. So there is a difference in predicted functional pathways based on the genes that we identified. Next slide, please. So in metabolomics, we're going to shift again here. So we've already looked at the genes, and I showed you there's a difference on the genes to identify certain bacteria that are present and the diversity. And the one comment I do want to make about the diversity that I forgot to say is that the more diverse our gut microbiota is, the better it is for us. So, and then I identified certain metabolic pathways that are identified with those bacteria. And now we're going to look at the metabolites. So those bacteria are busy digesting and breaking down all the food that's in our gut. And by doing that, they're going to make metabolites. And so those are things like you're familiar with, lactate and glucose and amino acids, all those are metabolites. And so this slide is showing you, again, the green is the individuals who said they consume protein supplements, and the orange are the non-protein supplement, is showing that there's a clear difference in the metabolite pattern within the fecal matter of these two groups. Next slide. So now the question is, well, which ones are they? And so we measured over 300 different metabolites. And what this graph is showing you is the VIP score. So these are the very important ones. And so these are the ones that come to the top. And on the left, you can see some of those listed, like allotonin and sucralose. I'm sure you're familiar with sucralose. And on the right, you can see that there are some colored squares there. And we have the no protein supplemented group and the protein supplementing group. And the blue means that it's higher in the it's low in that group, and the red shows that it's higher. So if you look at the very top one here, for example, allotonin, it is higher in the people who reported using a protein supplement. And actually, the first 5 here are higher in the ones that reported using a protein supplement. And sucralose is the artificial sweetener, and it's being picked up, obviously, in the fecal matter. And anybody can ask me a question about it, but I have no idea what's happening to it. Okay, so then if you take these metabolites, and let's go to the next slide, we can associate these metabolites with pathways too. So just like we did with the genes and we came up with metabolic pathways, we can do that with the metabolites as well. And so these are the top 5 pathways, most impacted pathways, and these are the highest in the group that reported using that protein supplement. And so what we're picking up is pyrimidine metabolism, glycolysis and gluconeogenesis, cysteine and methionine metabolism, purine metabolism, and TCA cycle. And I may have a few of you who are confused and it's like, what are all these things? So let me step back and give you a little bit of an explanation too, and why, who cares, and why this is important. So number 1, the pyrimidine metabolism, and number 4, the purine metabolism. These are nucleic acids that you find in RNA and DNA. And you remember, I think you know what RNA and DNA are. You know, that's the code for making all of the proteins that are in our body. And purines and pyrimidines are part of the nucleic acid that is present there. That nucleic acid is going to consist of a phosphorus group, a sugar, usually ribose, and then it's going to have purine or pyrimidine attached to it. So that's where pyrimidine and purine metabolism are important. And we're going to come back to that in just a minute. Glycolysis is the breakdown of glucose, and you all know glucose is the #1 energy substance in our body. That's what we rely on, our cells rely on primarily for energy. Glycolysis is breaking down that glucose without oxygen, and gluconeogenesis is then the production of glucose, new production of glucose. So you might take lactate and make glucose out of it. Cysteine and methionine are amino acids. And the TCA cycle, I know you all know what it is, maybe not in this fashion or called this, it's also known as the Krebs cycle, but this is the pathway that generates the energy in our cells. Okay, so those are the 5 most impacted pathways based on the metabolites. If you go back 1 slide, I'm trick Karen here. That's based on some of what you're seeing here. So we're picking up, for example, on top there, allotonin. So I just showed you number 4 on the next slide was the purine metabolism. So when you break down purine, which is a nitrogen base that was in part of that nucleic acid, which was part of DNA and RNA, but when you break down purines, you produce uric acid. I can go back to the next one, Karen. You produce uric acid. And uric acid, for those of you may be familiar, gout. You've heard of people who have gout. So what happens is they produce too much uric acid and they don't secrete it, and it then can get deposited in joints and things like that and cause gout. And we do not possess the enzyme to break it down to allotonin, but you saw that at the top of that list was allotonin. Our bacteria do. They produce they can take that uric acid and then they can break it down to allotonin. And allotonin is a very water-soluble substance that's secreted then. Obviously, we picked it up in the fecal matter. Can't tell you if it was absorbed because we didn't measure that. Next slide. So let's just put this all in some context and then I'll turn it over to Kristen. Okay, so we have dietary with protein, obviously, in our diet that we eat. It can come from a plant or an animal. Along with that is going to be nucleic acids as well. I mean, all living matter contains RNA and DNA. And so that's just going to come along with it because we eat living matter is no longer living maybe by the time we eat it, but anyway. Okay, so dietary protein can be then broken down into amino acids. A lot of it will be broken down in the small intestine and you'll absorb quite a bit of it, but not all of it. It's estimated that as much as 20% of the dietary protein that we eat actually makes it into the colon, where the bacteria can digest it. They're going to break it down into amino acids. They possess all of those enzymes. Those enzymes then can convert it into intermediates in the TCA cycle. And I should point out, this is purple. We also have allotonin purple here, and we have glycolysis that's purple. These purple ones, if you look over in the upper right there, these are coming from Metabolomics. This is information we got from Metabolomics. So we're trying to fill out these pathways based on all of the things that I told you about. And we also have glycolysis over here as well. I told you that folate was higher. And so that's coming from the diet information, but we know that the bacteria can convert some of these amino acids to folate products. On the nucleic acid side, then we can, the bacteria can break that down into, as I already said, those nitrogen bases, purines and pyrimidines. Purine can go into uric acid and then into allotonin. So next slide. So in summary then, oops, next slide. Okay, so we know that the group that reported taking protein supplements had a higher protein intake, higher fiber intake, more fecal metabolites and bacteria associated with nitrogen breakdown, and significantly higher levels of allotonin. Whereas the group that did not report using protein supplements, or report that they did not use protein supplements, that they had more genes associated with nucleotide metabolism and nucleic acid synthesis. So differences in diet and gut microbiome and metabolome suggest that protein influenced the gut microbiota, and we don't know what those health consequences are because we didn't measure that. Okay, Kristen, I turn it over to you.
Yeah, so you may wonder then, what's next? What's our next or our follow-up study? So the one that Lori just described was obviously a cross-sectional design where we compared two different groups. Our next study, we're looking at we're using a longitudinal design where we're going to, our main question is, how does the bacteria in our gut interact with the food we eat? And how specifically will supplementation with protein, or the addition of 50 grams of a protein-rich food each day for 2 weeks, impact those microbes in the fecal matter. So for this group, the participants we're looking for, we do not want them to make any changes to their diet besides the addition of that 50-gram protein food, 50 grams of protein. And we also don't want them to alter their activity in any way. Next slide, Karen. So here are our inclusion and exclusion criteria. And if you're listening, you may be a participant. So take note if you fall in that green category. We're looking for healthy, active adults over 25 years of age. So you do not have to be a self-described muscle builder per se, but we have some questions to see if you're meeting some minimal levels of activity. Exclusion criteria, just because of their effects on the gut microbiome, is diagnosis of cancer, irritable bowel disease, Crohn's disease, COVID, either current or long haul. And then a similar list as our previous study, use of laxatives, diarrhea inhibitors, pre or probiotics within the last week or the week prior to participation in the study. We also are limiting this to people who live within the contiguous United States just because of our need to have the fecal samples mailed. And then certain medications and antibiotics within the last 3 months would also exclude you. Next slide, Karen. And so here's our full study timeline. You can see there's a lot of action on the first day and then on the last day, day 1 and day 14, excuse me. We will ask participants to complete that International Physical Activity Questionnaire, the ASA 24, which is a diet recall tool, and then to mail their fecal sample, they'll be provided with a collection kit and a prepaid mailing address or label to send that, and with very clear instructions. And then they're provided with the 50 grams of protein, which they will take each day from days 1 all the way to 14. Each day, we also ask them to complete a daily food recall, indicating that they took that protein supplement. Halfway through, they repeat the IPAC. And then on day 14, you can see they repeat all those baseline tests of the physical activity questionnaire, diet recall, and one last fecal sample. Next slide, Karen. So if you have any questions, I think we have a minute or two, and we can always be reached via email. You can see all of our contact information there.
So the floor is open if anybody's got any questions. Any thought of doing it in adolescence? No, because adolescents, they add some extra layers of human subject need. So, will our audience be automatic registered ready for the study? Is that what the question, Doris, you're asking?
I think it's for attendance at the
Attendance
session.
here, yeah.
It has nothing to do with us.
But Jordana, Jordana, you asked a really interesting question, because as we tried to build upon the previous study and brainstorm for what to do next, I feel like we discussed every scenario of, do we change the population? Do we change the design? What do we change, and what do, it's that, the challenge of just changing enough to keep the science moving forward without, while still growing from what we learned in the past studies.
So you asked a very good question, Jordana, and they can interact. And you're using the word supplements. So does dietary, so we're looking at protein supplements. And actually we weren't looking at protein supplements. I guess we were comparing the two. They can impact it, particularly if they're replacing some other food groups and nutrients in the diet. So that can be a big concern. And I think there is some information about that out in the literature.
This is why we need a team name, because there's no shortage of potential studies that we've talked about and that you're bringing up right now.
Yeah, so Ed, the problem with us doing type 2 diabetics is just we would need some consult with an MD. We probably would need one single source. So in this case, we mail the fecal samples and they mail it back, so we don't have to have physical contact with the participants. There's lots of things. I would really like to study this in older people who have an increased protein need, and they also need to do resistant muscle development, and that can be done in older people. So we're talking about people who are 70 and older. And also that would help build their muscle. We also know that the gut microbiome changes as we age, and we talk about that. And so whether we could do some shifts that way, and whether changing the protein and fiber content could change the body composition as well. But I don't have any retirement centers that I can do it with. And I should say that for the gut microbiome, when we collected those fecal samples, the ones that were taking the protein supplements, there were only 4 of them out of the 22 who took a protein supplement the day that they collected their fecal samples. So most of them had not consumed a protein supplement. So we talk about them consuming protein supplements. That doesn't mean they do it every day. Most of the protein supplements that were consumed, because we do have a list of those, were whey. And most of them out there do contain whey. I'm glad you enjoyed it. Thank you.
I'm looking forward to your next chapter.
Yeah.
Yep, there'll be many. Several maybe.
Well, thank you to you, and thank you so much to everyone who's here today listening. I hope you learned something new about this world of research. It's a very fascinating study, and so great to see the findings that you brought forward. And again, I'm looking forward to seeing what happens with the next one. So thank you to you, and thank you to everyone else, and hope to see you this afternoon, or check out the site for our on-demand presentations. Thanks, everybody. Happy Friday.
Bye. Thank you all.